Okay, I think we can more or less slowly start. So I can start by giving you these. So while people are still joining, questions from yesterday? Anything unclear? Everything unclear? Something unclear? Yes? No, but I mean, the point is that these equations are fairly, I mean, there is no dynamics, right? It's statics, so it's basically almost always a linear system. So what I told you, what I told you so far doesn't involve any need of numerical stuff. What I'll tell you today, it needs, but I mean, it's fairly simple. But I haven't, so.
I mean, they are almost deterministic.
They are deterministic.
Because the R square of this curve, so it's almost 0.9 or 1.
Yeah.
Deterministic.
Yeah, I mean, yeah, but that's about the data, right? So that the data, I mean, there are like things that are, let's say, that are a little bit less trivial, which is how to analyze the data, but okay, it's a different story. The model itself doesn't require any numerical integration or simulation so far.
Yes, but it's nice to compare the data with the model.
Yeah, sure.
Put both data on the same graph.
Yeah, yes. Yeah, for that you need code, but it's like fairly standard, so it's not. Other questions? Yes?
So last year you told about this protein part we are dividing into two categories. One is this unnecessary protein that organism is building. I understand this point, but what's happening in the experiments? Can you just give a brief?
Sure. So what happens, so this is like about the overexpression of unnecessary proteins. So this experiment here. So basically, in a nutshell, what you do is that you so basically what you I have to remember how it's done. But basically, what you do is that you have a construct. So I'm not sure it's like this. I have to check. But here you have the protein you want to express. Let's say beta gal. Okay. So this is a protein you want to express. And so basically, the expression of this protein, it's regulated by another regulator. Okay. Which let's say. Regulates this, the expression. So basically you express this protein which regulates it. And so you can imagine that you express this gene, and this is like a transcription factors that positively regulates this. But this positively regulates this based on the presence of an inducer. Thanks. An inducer, okay? And this inducer, which binds to this and then let's say, I don't remember the details, but let's say that these, I think when it's bound, it's a repressor. It doesn't, it's always a repressor, and when it bounds, it doesn't repress. But basically what you do is that you can change this inducer. So IPCTG is the inducer, if I remember correctly. So I have to check. But this one, it's the inducer. So basically, you can tune how much of the inducer you put. And by tuning how much of the inducer you put, you tune how much you express of this. Okay? And so then, I mean, this is also, I guess, it's also fluorescent. So basically, you can measure how much you express, and you can control how much you express by changing the inducer. So you can do different experiment where you change the amount of the inducer, you can measure how much you expressed, and you can measure the growth rate. Okay? Did I answer? I have to check the details. I mean, the experimental details might be different, but the idea is that. Okay? Yes. Other questions? Yeah? Okay, so I haven't talked about that. I was just whoop. So this thing, I just mentioned it. I didn't say anything. This is just to say that the kind of model that we consider is a model where we said, okay, there are some nutrients, and then there are nutrients, then there are amino acids, and then there are proteins, and then there are the different sectors which control these fluxes, right? And so basically, let's say that you are in a bad nutrient. Then if you are in a bad nutrient, you're going to have a bigger P sector than an R sector. If you are in a good nutrient, you have a small p and a large R. Okay? Right. Now suppose that you do an experiment where cells are growing steadily in a bad nutrient. Okay? You have phi R as a function of time. Okay? Everything is stationary, right? In our picture, we are in a condition where everything is stationary. So this is constant. Okay? Now suppose that so this is bad nutrient. You have the growth rate, it's somewhere here. Okay? Now suppose that what you do is that you suddenly change the nutrient. Okay? You do an experiment where you remove the old nutrient and put the new nutrient immediately. Okay? Now, what do you expect? You expect there will be some transition. After, so here it's good nutrient. So something will happen here, and then you're going to end up here, right, with an higher growth rate. And let's say, sorry, it's the opposite. So this is like this, and then with a larger phi R. Okay? Do you agree? What I'm saying is, in a good nutrient, you have a larger phi R, a larger growth rate. In a bad nutrient, you have a lower one. Now, this is stationary. Suppose that you do it as a shift, so you change the condition, then at any point in time, a cell will have some amount of ribosomes. Right? So there will be some transition, and this transition will be dynamic. Right? So it will do something like this, or so this is one possibility, but something less trivial could even happen, so we don't know. Okay? So There could be other dynamics, right? So now, in our context, in our modeling framework so far, we cannot say anything about these dynamics because we are only considering at statics. If you want, this is completely equivalent to thermodynamics, right? Thermodynamics, if I do some kind of thermodynamic transformation, I don't know anything about the trajectory, but I know what happens at equilibrium, right? So if I wait long enough time, then I can say where I will be, okay? I mean, I can say what is the equation of state here, and I know that it has to be respected. This is the same. Now, the question that you can ask is, a cell, I mean, what's happening here is that you are changing the nutrient, right? So a cell, in order to do this transition, needs to have a way to regulate how many ribosomes it has, how many protein it allocates to metabolism, etc. Right? There must be a feedback, right, within a cell, some level of some kind of regulation that allows a cell to go from this state of ribosomes to this state of ribosomes, to this value of ribosomes, right? And this has to be done dynamically. So in principle, there will be an equation, right, where this will be equal to something, okay? And I know that when this is equal to 0, I get these three states, okay? Now, in writing this equation, what I have to put in is the regulation mechanism. So basically, there must be a feedback of something on phi r, okay? So the thing about PPGPP, which I don't want to describe, but this is the point I want just to make, because actually we're going to using it today, is there is a feedback, okay? So basically there is a molecule that it sort of sense, in some sense, how much free amino acids there are, and that controls the transcription of ribosomes. So how many new ribosomes are produced. So basically there is a feedback mechanisms between the pool of available amino acids and how many ribosomes you have, okay? And so if you change the condition, what you do is that you change the pool of amino acids. These produce, as a cascading effect, a change in the amount of ribosomes, okay? Now, there is a almost epistemological point here to make is, one could think the first things I'm going to do is to understand this equation and then derive the fixed point. What I showed you is that we had a good success by understanding something about the fixed point, and then sort of we can build, I haven't shown that, but we can build a dynamics out of it, okay? I mean, we can constrain the dynamics using what we know about the fixed point. Close parenthesis. Did I answer? Okay. Good. Other questions about yesterday, the day before yesterday, the day before yesterday? Okay. Blah, blah, blah. Okay, so we are here. Now, what I want to talk today is something more, I mean, it's an example of how we can use this to say something about the effect of antibiotics. And I'll see how much time I have, how actually this can be used to say something about the evolution of bacteria in presence of antibiotics. So, before I start with the experiments and the settings where I need to study, just one simple fact: there exist antibiotics, okay? Which I think you are aware of, because probably you all have taken it at least once in your life, some of you even now. So the point is, there are drugs, but there are like substances that affect the growth of bacteria. And affect the growth of bacteria by reducing how fast they grow or by killing them, okay? And our experience is that we take these pills when we are sick, and or where we don't want to become sick, and then this has an effect, blah, blah, blah. Okay, so the point is these things exist. Actually, one interesting thing is that these things exist and are typically substances that are produced in nature by other organisms. Okay, so one famous example, which was the way antibiotics were discovered, is penicillin, that is something produced by an yeast to kill bacteria. So this is like something that, antibiotics exist in the natural world, so it's not something that we made, and it's a new thing for bacteria of the last 100 years. It's something that bacteria are very used to, the existence of antibiotics, because are substances produced by other organisms to kill each other, okay? It's a nasty word also in that part, okay? So are toxins that different organisms produce to kill other organisms. And so it's something that is in the everyday experience of natural bacteria. Now, the point is, I don't know how much you are aware of this, but there is something you should be really scared of, is that there is a huge spread of antibiotics resistance. So basically, we have a finite amount of compounds we know, of drugs we know, of types of antibiotics, and bacteria can become resistant to these antibiotics. So if you expose, and perhaps you're going to see something more about this, but if you expose bacteria to a given antibiotic, sometimes they develop resistance, and so they are able to grow, and they are not killed by these antibiotics. And now this is a big concern, because we know a finite amount of antibiotics, and bacteria are sort of outsmarting them, and antibiotic resistance is spreading. So basically, there are many bacteria that are resistant to many types of antibiotics, okay? And so the problem is that if you get an infection from these bacteria and your immune system perhaps is low, then we don't have means to kill them, right? And we are used now to a world where we can control bacteria pretty easily, but we could be in a world where that's not the case, and that will be a big mess. Okay? Good. Questions about this? Okay. Now, the other point that I want to make is there are many types of antibiotics. Okay? So there are two main types of antibiotics, which here represents in red and blue. And so there are bacteria that kill, there are antibiotics that kill bacteria. So what they do is that when you expose bacteria to antibiotics, you kill a fraction of the cells based on how much antibiotics you put, and the resistance is evolved by the fact that bacteria are not killed anymore. And then you have antibiotics that don't kill bacteria per se, but they make them grow much more slowly, okay? So you have bacteria that they basically kill the cells, like they destroy the membrane, so they are killed, and others that interfere with the mechanism of action, and then they are not able to grow, okay? What's the difference? The difference is that in the second case, if then you have a bacterium that it's growing more slowly, and then you take it in an environment without antibiotics, you'll be able to grow as before, while in the first case, if you kill a cell, it will not be able to grow, okay? So experimentally, you can imagine of distinguish between the two very easily, right? Now, these antibiotics have many mechanisms of action, let's say, so they interfere with different processes that a cell has to do to be alive, right? So for instance, there are things that make bacteria unable to build the membrane, okay? Or there are the cell wall, so what is outside the bacteria. Or others that makes bacteria unable to produce proteins. either that interfere with the replication of DNA, so they are not able to replicate the DNA, others that they destroy the membrane, other that inhibits the synthesis of proteins by doing something to ribosomes. Okay? So we saw one of them, which is chloramphenicol, okay? And which is something that is actually not used as a, I mean, it's not something that is used to give to people, but it's an antibiotics that basically make ribosomes unable to translate proteins. Yes?
So, this science of bacteria killing, it's experimental, or you could simulate it with these equations?
What is the question? I didn't understand.
I mean, these facts that the bacteria, the body is resistant to the bacteria,
Uh-huh.
the DNA.
No, this is it's unrelated to models. It's an observation.
Okay,
So you,
experiments.
yeah, you can do billions of type of experiment. I mean, you can imagine to have experiments which are, say, in vitro. So you can, so let's say one line of evidence is that there are antibiotics, okay? And what you see is that there is an antibiotics that kill this type of bacteria. I know that you have an infection from this bacteria. I give you these antibiotics, and these antibiotics don't cure the infection. And so then I have to give you another antibiotics, and then it cures the infection. This is quite, I mean, something that is common. I mean, if you get like, I don't know, otitis, right? What happens is you go and, or conjunctivitis, right? You go, they give you the first antibiotics, the standard one. Then if it doesn't work, they give you another one, okay? And the idea is if it doesn't work is because the bacteria are resistant. Okay, and then typically you try, and so you have an order, and then you have, basically there are protocols on what you should use, and what you see is that, I mean, this is common experience that sometimes antibiotics don't work. Now, how do you know that this is because cells are resistant? Well, what you can do is that you can isolate the bacteria that cause the infection. So I take like some sample from your eye, if you have conjunctivitis, and the red eye infection. And I can take that, and then you can culture that bacteria in the lab, you can isolate them, and then you can see that those bacteria, which look very much alike other bacteria that are killed by the antibiotics, those bacteria are not killed or grow better.
So in this case, it is, the model is following the experiment.
There is no, I mean, for now there is no model. I'm just saying it's an observation.
Right? Yes, but for example, you have shown us the flask experiment with both aids. So I mean, with the model, it's calculating this growth rate, for example.
Yeah, you can, yes, you can, yeah, I mean,
In this case, the growth rate and all these
okay,
parameters
yeah.
follow the experiments. Once you do an experiment first, or watch, observe something, and then write a model.
Yeah, I mean, if you want, yes, you have always a model, but in this case, I mean, there is, the model, it's not a model of what's happening, of what is the mechanism of resistance, right? Then you could ask, okay, these cells are resistant, why? Okay, what did they mutate, right? They must have some mutation that makes them resistant. And this is something that we could do, and perhaps we're gonna do, but the point is, the first observation is resistance is widespread, right? And the evidence, it's also widespread. I mean, it's based on the fact that you can see bacteria that are supposed to be killed by antibiotics that are not killed. Okay? Okay. Other questions about this? Okay. Now, the point is, there exist antibiotics, and there are many types of antibiotics. And these antibiotics can either kill or affect growth of bacteria. And they have many modes of action. They can interact with the processes happening inside a cell in many different ways. Now, what we are going to focus on today are antibiotics that inhibit the synthesis of proteins. And so in particular, antibiotics that affect the functioning of ribosomes. Okay? Because this is something that falls well with the model framework we are building. Okay, so we are focusing on these antibiotics. Now, suppose that you want to that you are an experimenter and you want to know what is the effect of these antibiotics on cell function. Okay? So whenever you have a drug or a substance or a molecule or something that affect some function of an organism, there is a typical first things that you need to know. Right? Suppose that I have a drug, okay, and I know that this drug or a molecule, this molecule will affect the growth of something, okay, or will produce an effect on you if you get this molecule. What do you think is the first thing you need to know before giving it to someone? Well, you need to know it's too dangerous, but we know that something is dangerous based on the effects, but also the dosage, right? So one key question I have to ask is, suppose that I have a molecule that I know it produces an effect. I want to know how big is the effect based on how much I give it, right? This is true for whatever you want. I mean, it's true for drugs that we take. It's true for antibiotics that we give to bacteria. It's true for everything, right? We want to know what is the scale, right? So there must be a typical concentration, let's say, below which, and if we are much below that, there is no effect. If we are much above that value, there is a big effect. Okay? So this is the first thing we need to know whenever we want to really use, yeah. No, I'm talking about very abstract thing, right? So then you can ask, your question is related to if I give something to an organism, this could be bacteria or humans, then there is another question on whether how much heterogeneity is important. Okay, and this is another topic if you want. So there is also that concern, but for now we say, let's focus on the simple case of homogeneous population. Okay? So what it's always done, basically, what is typically done, is that you have some function, okay? And you measure this function. This could be, for instance, growth rate, right? So in our case, could be growth rate. So you have the growth rate. And let's say that this is the concentration of the drug. Here you have 0, okay? And I want to know what is the growth rate over the relative to no drug, right? So this is 1. So if I give no drug, I have 1, okay? And then I ask, suppose that I increase the concentration. For instance, the growth rate, I expect it to decrease. This will decrease. And this can have whatever form, right? But let's say it will go something like that, okay? And a typical parameter that is studied is what is the concentration where you have, in this case, half of the initial growth rate. So this is called inhibitory concentration of 50. Okay? Now, this is a general thing you can do, and you can imagine that this can be the growth rate, if you're interested in growth rate as we are, but this could be, if you have a drug that kills bacteria, this could be the percentage of cells that are alive, right? So you put a given concentration, you have the drug act, and then you measure how many cells are able to grow in a medium without antibiotics, and you measure what fraction of cells you killed. And you measure these at different values of concentration, and then you have the typical value of the concentration when you kill 50%, right? And this tells you what is the scale, right? The scale we wanted to know. So if you are much below this value, you know that there is no effect. If you are much above, you have a huge effect, okay? And so depending on what is your goal with this drug, you can sort of balance where you want to be with this, okay? Is it clear? Good. Now, let's focus on the drugs that affect antibiotics. What we can do is that, as before, what we can do is, you remember that we were measuring, so we had the effect of, we have the, for instance, different chloramphenicol concentration and different growth rate, we can produce the same plot, okay? And so, for instance, this is one drug, okay? Actually, yeah. So this is one drug, and what you do is that you measure the relative growth rate. So this is the growth rate when you don't have the drug. And focus just on one color for now. For instance, the red one, you are here, and then you increase the concentration, right? This is a concentration, is how many micrograms you put per milliliter of your culture. What you do is that you increase the concentration, and you see that the growth rate of the cells decreases, right? So what this point represent an experiment where you put your bacteria and you put some concentration of the drug, right? And then you have this bacteria growing over time, and you measure the slope, and this is lambda, okay? And so you want to know how lambda depends on the concentration of the drug you put, okay? And so here you put, in this notation, lambda naught is the growth rate when the concentration is equal to 0, okay? So focus on one color, okay? Now, what you can do then is to say, I can repeat the same experiment with different nutrient, right? So I can do an experiment where I put, so with different nutrients, and different nutrients corresponds to different initial growth rates, so to different values of lambda naught, right? So for instance, I can do this experiment where I give the red one, where I give as a food glycerol, okay? And so the cells are growing with lambda naught, which is the growth rate is 0.4 per hour, okay? But then I can repeat the same experiment where, with the same concentrations, I give as a food glycerol plus amino acids. So lambda naught is equal to, is twice as before, okay? And now what I have is that I have, all the curves have to start from 1, because with 0 concentration, I sort of normalize the growth rate, but then will decrease, and the decrease is different depending on the nutrient, okay? Is this clear? Good. Now, here you have two types of experiment. You see these are glycerol with different medium, and then you have also glucose with different medium, right? And so here is reported only the one for glycerol. But then what you can do is that you can say, okay, now I can plot the value where these, where the growth rate is 50%. So these I50 that I was talking about, right? So I have this concentration, okay? And so I can ask how this concentration, this inhibitory concentration, depends on the growth rate without the drug, okay? And so what you see is that for this drug, it goes up, right? So if you are in a nutrient which is better, you have to add more higher concentration of the antibiotics to achieve the same relative effect. Okay? So to say this in a different way, if I'm growing at 0.4 per hour, I have to add 0.2 microgram per milliliter to grow at 0.2, so at half of the growth rate. If I'm growing twice as fast, I have to add more to grow half as before. Is it clear? Questions about this? Yes?
Each time we're trying to calculate the concentration and the relative growth, we are taking a different sample?
Yes, so these are different experiments. So you do one experiment where you have cells, and then here you give like, let's say, glucose plus amino acids, okay? And then you can do another experiment where you have the cells, and then you have just glucose, right? And then you have a larger concentration of the drug. Okay? So you do these, separate, you grow them, you measure the growth rate. Okay? Yes?
In the left graph, what is the arrow represent?
The arrow? No, the arrow is just to say that you see that these curves ordered by the growth rate, so red, blue, and green, it goes up. Okay? So the fact that this arrow goes up is because this goes up, right? Okay? Good. Now what we can do is that we can repeat this with another drug, and we see a different shape, right? There is a different shape. There is a different value because it's a different drug. But so before the scale, I can go back and forth so you can see. So the scale was 0.2. You see the red point for IC50. Now it goes down, okay? It's 0.1. So you see that you have a different scale. So for instance, with this drug, you need a much lower concentration to achieve the same effect. Right? And you also see that the curves are somewhat different. Okay, they have a different shape. In this case, you see the same effect, right? You see that this IC50 goes up with the growth rate, okay, as before. So you see that curves are ordered this way. Okay? Questions about this? Yes?
The above is the name of the drug.
Yes?
And we are changing the type of bacteria? Or what?
No, we are changing the nutrient. So each color, so we are changing two things, right? As here. We are changing the nutrient
And the drug.
and how much the drug and how much of the drug. Okay? So each plot is, you fix the drug, you change the concentration and the type of nutrient. Clear? Other questions? It was 3 milliseconds first.
If we compare this graph with the last one, the scatter of that points is more it's lower in another one than
So the x-axis is the same. Don't know why. Okay. If you look at the x-axis, I don't know why. Okay. The x-axis is the same, right? The y-axis is changing. Yeah, I mean, we are going to go there, but the point is there is something about the drug which is different, but this is, I mean, these are different molecules. They have different properties, so it's expected to have different things, right? One obvious difference is that they have different concentration. But this doesn't change because it's an experiment without the drug.
But it's more likely linear.
We're going to go about the, the story is not, yeah. But the point is just to say, I want that it's clear what we are measuring here, okay?
So I was wondering, this relative growth rate, it changes this profile dependent on this.
Yes.
So IC50 is like a good measurement, or do you shift this like?
No, I mean, it's not the full story, right? You see that the shape of these curves is very different from before, right? And IC50 is just something that tells you what is the scale of the concentration, right? So you have, this is difference because this axis is changing. So it's changing what numbers are here. I could have a drug where here I have 0.01. It's a very powerful one. Or I could have one where I have 1, right? So IC50 is a scale as a unit of a concentration. So it tells me what is the scale. But then also the shape of this curve is different. We're going, yeah.
So the question is like, if your profile is very slowly decreasing, I mean like, so IC50 might not give you a good measure like if.
No, the point is you could have a profile which is like this, okay? And in the same scale, you have something like this, right? And here you have, I don't know, IC50 which is here. And here you have IC50 which is here. But it could still be true, in principle, that if I plot the concentration divided by IC50, then the two curves are the same, okay? This is not the case, right? If you look at the shape of these curves, they don't look very similar. I mean, they have some similarity, but they are not the same, okay? Okay? Other questions? Okay. These are two drugs. You can try another drug. And what you see is you have other shapes. But in this case, you get the IC50 decreases with the growth rate. Okay? So you see that in this case, if you were growing at a faster growth rate, you need less drug to achieve the same proportional effect. Okay? Huh?
Why does this happen?
We have 45 minutes to answer. Okay? Then you try another drug, chloramphenicol, which we saw. Again, it decreases with the okay? So this is an interesting observation, right? We saw 4 drugs, and we saw 2 different, we saw that for 2 drugs, the inhibitory concentration, this IC50, increases with the growth rate without the drug. In other 2 drugs, it decreases, okay? So what I want to show you now is that we can, using sort of simple models and what we have discovered, we can explain these. I mean, you could say, okay, there is no single mechanism. So I could describe these drugs with one type of models and these drugs with another type of models. I want to show you that no. These are 2 expected behavior from a very simple model with the same assumptions. Okay? So what we want to reproduce is this phenomenology. Okay? Is it clear what we have to do? Good. Questions? Okay. So let's try to write a model. So the model we want to write is a model that takes into account 2 main ingredients. Okay? One ingredient is something about what we discovered and discussed in the previous lectures. And the second ingredient is how antibiotics affect the functioning of ribosomes. So these are all antibiotics that affect how ribosomes work. Okay, and we want to describe how they act. Remember that what we want to characterize is how the growth rate depends on the concentration of antibiotics. So we'll need to have explicitly a parameter, an input, which is how much antibiotics we put in the culture. Okay? So we're gonna, so how do these antibiotics work? So as a first approximation, so we have ribosomes. Now I'll use R instead of PR because here we want to describe concentrations instead of mass units, but it's the same thing. They are proportional to each other. Okay. So we have ribosomes. Okay. Now the idea is that when you have antibiotics, these kind of antibiotics, what they do is that they bind to ribosomes. So, okay, so 90% of what we do is things that bind and unbind. Okay, so antibiotics, they bind to ribosomes, and when they bind to ribosomes, they make them not working. Okay? So what we have is that we have that ribosomes can be in two states. Okay? So can be bound to an antibiotic or can be unbound to antibiotic. Okay? Or actually can be, if you want, inactive, but those we don't consider. Okay? So these ribosomes, so there is a transition rate of going from bound to unbound. Okay? And so the rate at which you go from bound to unbound is some K off, okay? And so it's the rate at which the antibiotics unbinds to the ribosomes. And then you have a rate at which you become bound, which is proportional to K on times the concentration of the antibiotics. Now, this concentration of antibiotics is the concentration inside a cell. Okay? So it's the concentration of antibiotics inside a cell. Now, the concentration of antibiotics inside a cell is not equal to the concentration of antibiotics outside the cell. Okay? So there is the way antibiotics enter in the cell is not by passive transport. They are actually transported through other mechanisms in which cells transport things from outside. So there is an active importing and exporting of antibiotics. Okay? So this means that there is a concentration outside the cell, okay? And this is in general different from the concentration inside the cell, which I'll just write with A. Okay?
Excuse me, but
Yes.
inside a cell, you mean inside a bacteria, yes?
Yes, inside a cell. So you see here the scheme. So you have A, unbound ribosomes, bound ribosomes, and the concentration outside. Okay? And you will have some influx and outflux. Okay, so what we want to do is we want to write equations. And now I'm going to write equations that are dynamics and then study the steady state. So what is the dynamics? Well, I want to write an equation for the internal concentration of antibiotics. Then I want to write an equation for the bound ribosomes. And then an equation for the unbound ribosomes. Okay? Good. So how does, So in all these terms, these are concentration. Cells are growing exponentially. So in all these terms, I have a term which takes into account dilution by growth. So if cells are growing and somehow I stop importing antibiotics, the concentration will decrease because the volume increases. So in all these terms, I have minus lambda A, minus lambda RB, minus lambda RU. We saw this term 2 days ago, okay, when we study concentrations. So if the number is fixed and the volume is growing, the concentration decreases by this factor, okay? So this term is always there, where lambda is the growth rate, okay? Let me write what is our goal. Our goal is to understand what is lambda over lambda naught as a function of AX. Okay? This is what we want to find. Right? And how this depends on lambda naught. Okay? So this is the result we want to achieve. Is this clear? Okay. Good. So we have this term everywhere. Then for the concentration, we have effective flux of the antibiotics. Yes.
You said those are decreasing, the concentrations are decreasing because the volume is increasing.
What I'm saying is if the number, suppose that you have a molecule and the number is fixed, cells are growing exponentially. Okay, the concentration decreases because the volume is increasing, but the number is fixed.
But the unbound ribosomes are going to increase too.
No. There are other terms. This is not the end of the equation. There are other terms because then there are other reactions. But let's say there is always a term which is related to this dilution. So if you write an equation for the number of something, okay, which is f of n, if then you have the concentration of that something, you will have that dc, right? The T will be equal to f, well, let's say some other, let's say this is capital F, this is small f, will be f of c minus lambda c. Okay? I'm writing this term. Okay? Then we're also writing this. Okay. So, good. So this term, we have a term which is the effective flux. So it represents these steps here. Okay? The fact that things are coming in and going out. Okay? And so this J, so for instance, this J X, for instance, is equal to some rate P in AX minus P out of A. Okay? So the way this changes is the amount of concentration inside a cell increases because with some rate you import something from outside, proportional to the concentration outside, and with some rate you take something inside and you put it outside. Okay? Good. Then what we have here is that the number of available molecules decreases when this binds to a ribosome. Okay? So this decreases with a function of Rb, Ru, A, which represents this binding. Okay? Now, the number of binding one increases with the same function, and this decreases with the same function. Okay? And this function it's equal to what? It's equal to, how do you increase the number of bounds? You increase them with a rate which is k on A times their bound, which is the rate at which you here, and you decrease it with the rate which is K of R bound. Okay? Good. Is there any term that we are missing? Eh? The answer is yes. Eh? Degradation is there. Well, you could have active degradation, which could be a term additional to this, but we neglect it. There is another term. K-off is there. So this term here, it tells you what is the rate at which you change the amount of bound ribosomes, and you increase it with this rate, and you decrease it with this one. Okay, so this is the effective change.
I think we need a term that takes into account the increasing number of the elements with lambda.
Exactly. Yes. So if you are like this, you see that the total amount of ribosomes, right? If I just wrote like this, I sum these two terms, so I can get dr dt. Okay. If I just sum these two, what I get is these two terms cancel, and I get minus lambda r. Okay? So why do I get this? Because I haven't included any mechanism, because cells are producing ribosomes while growing, right? So the cells is not just stick with the ribosome it has to keep producing ribosomes as it grows. Now the question is, how many ribosomes do you produce? Now this is non-trivial, right? Because you could say the amount, you could say, you know that the amount of ribosomes you produce depends on the growth condition. Okay? So here you have a term which we say it's a function of the growth rate. Okay? And now, do you agree that you have this term? And so this term has to appear somewhere. Now, when you produce ribosomes, you produce them unbound to the antibiotics. So this term is here. Okay? If you want, when I wrote the equation at the beginning answering the question about PPGPP, what I said is you have these mechanisms that sense the amino acids and produce ribosomes. That mechanism is this S of lambda. Okay? So what we're saying is there are antibiotics that binds to ribosomes, but still you have this mechanism in place, which you didn't touch. And so whatever you were sensing, you are still sensing it and produce ribosomes. Okay? So these are the equations that we have to deal with. Now, everything it's sort of specified except this lambda here, this S of lambda. Okay? How do we specify this S of lambda? We know that whatever is this S of lambda is such that this condition holds. Right? This is what we know. Right? We know this steady state. So you can derive that form, but basically what we want to know at the end, so we have this equation, we have some S of lambda. What we want to find are the steady states. Okay? We care about steady growth, so we want to find what happens when all the derivatives are equal to 0. Okay? Okay? So we want to find the fixed point of this equation.
Sorry, before you go on to that, why not, do we have anything here that talks about the concentration of the nutrients?
It's not explicit. The concentration of the nutrients comes into this thing here. okay, in some sense, because we know that the rate at which we produce ribosomes depends on the nutrient, right? Because we know that if you change the nutrient, you're moving on this line, okay? So it's inside that factor.
Excuse me.
Yes.
Could you please repeat why you want to calculate this fixed point?
Because we care about balanced exponential growth. And so in balanced exponential growth, we know that these processes are in steady state. Okay? So the experiment that we did was an experiment that was done in balanced exponential growth. So whatever we find there, it should correspond to the steady state of this equation. Okay? Now, the steady state depends on the choice of this lambda, on this S of lambda. And what we want to impose to that S of lambda is the following condition, is that the sort of the first and second growth law. So the first growth law is telling me that the growth rate, okay, so I have this condition plus, let's say, fix S of lambda and lambda, such that the first growth laws applies, which is lambda is equal to what? Is equal to k t, okay, the translation rate. What is translating? Only the unbound ribosomes. So what we know is that this has to be equal to Ru star minus R naught. Okay? So this is the result of the first growth law. So what we know is that what is translating are inactive, unbound ribosomes. So the growth rate must be equal to, proportional to the unbound ribosomes. Okay? Good. And then what we know, okay, is this law here, which is something about all ribosomes. So this is one. And so we know that the total amount of ribosomes has to be equal to some R max minus lambda over Kn. Okay? Where the nutrient enters. Do you agree with these two things? So we are imposing these two. Imposing these two imposes these two conditions, which is based on what we know from the previous lectures. Effectively, what it does is that it fixes the form of S of lambda, and it fixes the value of lambda. Okay? Now I want to re-express this thing here using the fact that the growth rate in absence of the drug is equal to R max minus R naught Kn Kt over Kn plus Kt. Okay? And so I can express Kn, which appears here, as a function of this lambda naught. Okay? And so what I get if I do that is this R star, sorry, it's equal to R max plus lambda over kappa t minus R max minus R naught lambda over lambda naught. Okay? So I have this equation, these two equations, and then I have the ones that I get from there. Okay? And the ones that I get from there, it's 0 is equal to minus lambda A star plus P in AX minus P out A minus K on A RU minus K of RV. Okay. And then I have the second equation, which is 0 equal to minus lambda RB star plus K on A star RU star minus K of RB star. And then I have another equation which half of you cannot read, which is going to be minus, well, in principle, I have this other equation, okay, which is something that depends on S of lambda, okay? But I will not write this equation. I will not write this equation because I fixed S of lambda by using these two, okay? So I could write this equation and I could use this equation to derive what is the value of S of lambda, but I will not write it, okay? Questions about the math? Yes?
The antibiotics external concentration, it remains constant?
What?
The antibiotic external concentration.
The external concentration is a parameter, right? This is exactly what we want to derive. Okay? Yes?
The S of lambda, we said like it's kind of depending on the nutrients that we're adding to the system, but now we're not saying that.
No, no, we are saying that.
But like if we said that we kept it a fixed point because we kept lambda at a fixed point.
Lambda is a fixed point, but it depends on the nutrient. So what I'm saying is you have here a dependency on the nutrients. Okay, so here you have, in some sense, two unknown things which we are fixing with this. One is what is the shape of S of lambda, and one what is the value of lambda. Okay? We are fixing both of them by using these two. Okay? And you see that in these two conditions, there is a dependency on the nutrient, on this kappa n, or if you want a dependency on lambda naught. Okay?
Can you repeat, please, briefly the idea of these two conditions?
These two conditions?
Yeah.
These two conditions are the conditions we learned here. So there is one condition we learned from experiment, which is the fact that the total amount of ribosomes decrease linearly with the growth rate when we add antibiotics. And this is the second one. So that the total ribosome concentration decreases linearly this way. Okay? And then we know that the growth rate is proportional to how many ribosomes are translating times their translation speed. Okay? And this is the first one. Okay? So what we have is, yes?
Can you repeat why we got to the last equation?
The last equation is because the last equation we have, we have this quantity here, okay, which is what determines the total amount of, let's say, it determines how many ribosomes we have, basically. And now I said, well, what is this S of lambda? How do we know what it is? We can fix it because we know what is the total amount of ribosomes. So, and we know it by this. So you could obtain an expression from S of lambda, if you want. So if you want, this becomes a variable, okay? Right? And how we fix this variable is we fix this variable by, for instance, using this equation here, okay? Now, I can write that equation and then write the value of that variable explicitly. But in this context, I don't care about the expression of S of lambda. So I don't write that equation, and I'm happy with that. Okay? Yes?
The last equation, not
No, it's minus, plus k of, thanks. Thank you. Okay, so what I have here, it's 4 equations for 4 variables. What are the 4 variables? Ru, A star, the internal concentration, Rb, and lambda. Okay? So we have 4 equations and 4 unknown things. And the one we care about, it's lambda. Okay? So what we want to find is solve this equation to get an expression of lambda as a function of Ax. Okay? Is this clear? If this is clear, what follows is simple algebra. Okay? So I will not do the full derivation because it's sort of simple, but I'll sketch it out. So I remove this. And so I'll rewrite the equation we need to solve. So what we need to solve is lambda equal to kappa t Ru minus R naught. I remove the star also because then we have the second equation, which is R, which is Ru plus Rb plus R naught. Yes. No, these are Ru plus Rb. Which is equal to R max plus lambda over kappa t minus R max minus R naught lambda over R naught. And then I can write this equation here, which is Rb equal, I can directly solve it, kappa on A Ru over lambda plus k off. Sorry, this was R new. Here there was an R naught. Okay? Ru minus R naught. Okay? And then I can, you see that this term appears in both the equation. So I can sum them and I get lambda A plus Rb equal to P in Ax minus P out A. Okay? So you have this system of equation that you need to solve. Okay? So now it's boring things of changing stuff into each other, and after some algebra, and what we want, I mean, the goal of this is to obtain lambda. Okay? So what we care about is to obtain an expression for lambda, an equation for lambda. So after a while, what you do, you obtain an expression that I'll write down because it's like the one critical step. So you do this, all these steps and you get an expression which looks like 1 minus lambda over lambda naught. Yes. 1 plus kappa t over k on times lambda over lambda 0 squared plus lambda over lambda naught kappa t over k on over lambda naught k of. You might wonder why I'm writing this, but this is because this is a critical step of the calculation where we're going to do some simplification. Plus kappa t p out k on over lambda naught squared k off k on equal to lambda over lambda naught I just realized something. Sorry. Just realized that this is, sorry, I realized that this is not the expression I wanted to write, and I was copying something not updated. Okay, so okay, so this is the expression you get, which is an equation for lambda. Okay, so basically when you remove all the you have all the solutions, so you remove all the other terms. Now what you see is that this equation depends on all the parameters that we had. So it depends on the rate where you go in and out and the external concentration. Right? You also see that this is a cubic equation in lambda, so it's something that is not super easy to solve, but I mean, it's not something that you can solve with pen and paper. But then you can also see that if you look at dimensional analysis, you can see that you have, in particular, this term, which has the dimension of the square of a growth rate, because all of these are rates. Okay? So you see that these two rates cancel, and then you have this rate square. So I'll define this as lambda bar squared. Okay? And you can also see that this term So this term is the dimension of a rate to the square, okay? And this term as well, okay? So I can rewrite this term as lambda times this lambda bar, the external concentration over something that I call A50 bar, okay? Where A50 bar it's equal to R max minus R naught over P in lambda bar. Lambda bar it's here. And actually I put a 2 here, which becomes a 2 over here. Okay? No, I did it incorrectly because, of course, I was looking at the... Oh, it's correct. Okay, perfect. Okay? Now, this term here, using this definition, you can define it as KT over K on times K off plus P out over 2 square root of K off P out. And now I'm going to make a simplifying assumption, which is the fact that I'm going to assume, so this is like basically, we started from here, we get this equation for lambda, and then we redefine parameters. So we identify 2 parameters, one that has the unit of measure of a concentration, and one that has the unit of measure of a growth rate. Okay? And now we have these 2 other terms which we cannot express easily in this. But what we see is that both depends on this ratio kappa t over kappa on. Kappa t over kappa on. So this ratio here is a ratio between how fast antibiotics binds to the ribosomes, the rate at which it binds to the ribosomes, and its ratio with the rate at which ribosomes translate a protein. Okay? So the idea is that the antibiotics binds with a rate to the ribosomes much higher than the rates it takes for a ribosomes to translate a protein, right? Otherwise, the antibiotics will be basically ineffective. It means that what a ribosome has to do, it's like it can do it without being affected by the antibiotics. Okay? So our assumption, which is like also something that is sort of expected, is that KT is much lower than K on. Okay? And so under this assumption, I can neglect this term. And I assume that this is order 1, right? This is like something related to, it's basically the mean of K off and P out over their geometric mean. So it's something that is reasonable that it's order 1. I mean, it's not exactly equal to 1, but it's not something very small or very large. But this is very small, so I'll neglect this term here. Okay? So under this assumption, what I get is 1 minus lambda over lambda naught, lambda square plus, sorry, here there was, this is defined lambda over 4. It doesn't change anything else. It's just the definition of lambda bar plus lambda bar squared over 4. Equal to 1/2 lambda bar A over A50 AX. Okay? Now to make both the sides adimensional, I'll divide by this lambda bar squared. So I get lambda bar squared. Here I get 1. And here I get lambda bar squared. Okay?
Dividing both sides by lambda bar squared.
Yes.
So in the right hand, I should have
Lambda bar. Yeah, sorry. Yes, correct. Okay, so now you see that everything is adimensional, right? So that you have this ratio, this ratio, and this ratio. Okay? Now, since it's the end, I want to just give you, we'll continue on Monday with this, but I just want to give you a glimpse of why this is something that we are going to like. So what is I50? Okay, so the value A50 is the value where lambda over lambda naught is equal to 1/2. Okay? So what I can get is the value A50 here, AX equal to A50 when lambda is equal to lambda naught over 2. Okay. So now if I take this expression here, I can divide by this term here. And here I get 1 over lambda, I have 1 minus lambda over lambda naught, lambda over lambda bar, plus 1 over 4 lambda bar over lambda equal to 1/2 AX A50. I can take these two and put it here. Now, if I substitute lambda equal to lambda naught over 2, and I substitute AX equal to A50, what I get is that A50 over A bar 50, which sets a scale, okay? It's equal to what? So here I have a 2. Now lambda is equal to lambda naught over 2, so this is equal to 1. And here I get lambda lambda naught over lambda bar with a 2. And here I get a 2 which simplifies this. So if you substitute, what you get there is lambda naught over lambda bar plus lambda bar over lambda naught. Okay? Do you agree with the calculation? What I did was to substitute lambda equal to lambda naught over 2 and AX equal to A50, which is the value corresponding. Okay? And what I get is this expression here. Why this expression is nice? Because what you see here is that this function here is a function of, it's a function of A50 as a function of lambda naught over lambda bar. It's something that does like this. Okay? So if lambda naught is smaller than lambda bar, if lambda naught is smaller than lambda bar, this term dominates and the function is decreasing. If lambda naught is larger than lambda bar, this term dominates and it's increasing. Okay? So what we see is that depending on how big is lambda as a function of lambda bar, this inhibitory concentration can decrease with lambda naught or increase with lambda naught. Okay? And now, what is cool about this expression, and this model, is that we started with all of these. Basically, we need to put ourselves in this condition, but then we express everything in terms of these two drug-specific parameters, which is this lambda bar and this scales A50 bar. Okay? So we express all the parameters inside these two effective parameters. And we see that depending on what is the growth rate compared to these effective parameters, we can have a decreasing behavior or an increasing behavior. Okay? So what we are going to do next time, I was undecided what to do, but now I'm not undecided anymore. So what are we going to do on Monday is to finish this and to show you how then this can be used to understand something about resistance. Okay? And with Monday, we conclude our adventure in microbial physiology. Okay? Good. One last thing. So since you are writing all the things, I put some notes on my website. So if you go jacopogrilli.github.io website notes /Spring College 2025, and you have some notes for the lectures. So 3, you have the slides and some calculations, okay? And the references at the end, okay? So, whoop. Good. So you have, if you want to read something during the weekend, you have what to read. Yes?
In the GitHub site.
It's, I'll write it here.